This is Module 3 titled Transcription. In this video, Introduction to Transcription, I'll describe some properties about gene expression, which is another word for transcription, that are common to both prokaryotes and eukaryotes. First, I want to explain the importance of RNA in expressing our proteins. One way to do this is to look at what happens when RNA is not able to be made. Remember that all of our genetic information is stored in our DNA molecules, but that protein cannot be made from DNA directly. It must first pass through an intermediate, which is RNA. So DNA serves as a template to create RNA, and RNA serves as the template to create protein. The death cap mushroom, which is Amanita phalloides, causes death by inhibiting this process. This is a mushroom that's fairly common in Europe and looks a lot like other commonly consumed mushrooms as well. So it's very closely similar, physically similar to edible mushrooms, but it is a very deadly poison. In fact, it only takes about half of a mushroom cap to fully to kill an adult person, to poison an adult human. And of all of the accidental poisoning events by mushrooms, this mushroom leads the pack in the most commonly the mushroom most commonly associated with poisoning. It produces a toxin that binds to RNA polymerase, which is the enzyme responsible for creating an RNA molecule. When this toxin gets into the RNA polymerase enzyme, it stops its ability to bind to our DNA and create an RNA molecule. Without RNA, you can't make any essential proteins. Without RNA, you can't regulate many genes. And so if you can't do any of those things, death is fairly certain and fairly quick. It's actually really interesting when you look at the history of RNA and its discovery, because lots of evidence suggests that RNA was actually the original genetic material over DNA. There's a couple of different things that support this idea. RNA and DNA are very, very similar in structure. So they both have nucleotides. They're both have a sugar base, a ribose or a deoxyribose. They both contain nucleotides joined by phosphodiester bonds, and they both can take on secondary structures. The major difference between DNA and RNA is the presence of a 2' OH group on the 2' carbon of the ribose sugar, or the deoxyribose sugar. So that's not present in DNA, and it is present in RNA. And that actually increases the stability of DNA over RNA quite a bit. And so RNA, when it's single-stranded, is very easily degraded by enzymes. DNA is much more stable. But all of the information required to make a protein is contained within an RNA molecule. Offering further evidence is the fact that some RNAs are catalytic and are capable of catalyzing chemical reactions. In order for a molecule to, or a storage molecule, an information molecule to be translated into a protein, there needs to be some kind of chemical reaction that drives that process. And ribozymes is one example of an RNA that is catalytic, meaning it can catalyze, so it can actually break phosphodiester bonds and remake them. So this is an RNA molecule, just a piece of RNA that actually has enzymatic activity. And so it's long been thought that RNA, we actually live in an RNA world, that RNA actually started out as the original hereditary material, which was made possible by its catalytic activity and its ability to store information. However, the evolution of DNA greatly improved its stability, and it was not so easily degraded anymore. And so this improved the cell's ability to live longer, to last longer. So let's look a little bit at the structure of RNA. Typically, we think of RNA as having only a primary structure, which is a single strand of ribonucleotides. But RNA is actually capable of folding into many types of secondary structures, very complex secondary structures also. And it's these secondary structures that give some RNAs their catalytic activity. So this is the primary structure of RNA. So here's our phosphodiester backbone, the 5' end with the phosphate, and the 3' end with which would be an open OH group if this wasn't connected to another nucleotide. Remember that RNA contains uracil instead of thymine, and that RNA has a hydroxyl group on the 2' carbon that is just a hydrogen atom in DNA. So this is the primary structure of RNA. It's sequence, it's long sequence. But RNA is created using complementary base pairing to its DNA target. And because of that, and a lot of the same nucleotides are used also, with the exception of uracil, because of that, nucleotides within an RNA molecule can actually hydrogen bond with itself and form these complex structures called hairpins, where there are regions of complementary base pairing separated by regions that do not base pair, typically base pair. And so we can get large complex RNA structures through the formation of these hairpins. And those hairpins sometimes have activity. In fact, those hairpins are essential for the function of a specific type of RNA called transfer RNA. Transfer RNA is made in both bacteria and eukaryotes, and it helps to incorporate amino acids into a growing polypeptide chain. We haven't talked about this yet, but transfer RNA helps with the process of translation, or using RNA as a template to create protein. Ribosomal RNA also does this. Ribosomal RNA is also found in bacteria and eukaryotes, and this is a smaller piece of RNA that's part of a larger enzyme. So the ribosome, which is a combination of protein in RNA, consists of a small section of ribosomal RNA, which helps it do translation, or using RNA to create as a template to create proteins. Messenger RNA is also common to both bacteria and eukaryotes, and messenger RNA is essentially what you get when you're trying to make a protein. So messenger RNA is the precursor to a protein. There are lots of other regions of the genome that will be transcribed into RNA, but don't themselves get translated into proteins. These are typically found in eukaryotic cells, with one exception, which is the CRISPR RNA, which is only found in prokaryotic cells. So things like small nuclear RNAs, small cytoplasmic RNAs, microRNAs, and small interfering RNAs, all of these different types are different forms of regulatory RNAs. So they're involved in processes like processing the messenger RNA for creation into a protein, or translation into a protein. Small nucleolar RNAs are responsible for processing ribosomal RNA for integration into its enzyme. Small interfering RNAs trigger degradation of other RNA molecules, which helps to regulate gene expression. This is actually how we can turn off the expression of a protein in the body, is by creating these small interfering RNAs, which bind to messenger RNAs and chop them up into bits, and so they can no longer be used to make protein. And so there's a wide variety of different shapes that these RNAs can take, and because of that, they have a wide variety of functions as well. They can be small, as is the case with these small guys, micro and small interfering RNAs. They can be much larger, as is the case with messenger RNA. They can take on complex secondary structures like transfer RNA and so on. Regardless, all of these different types of RNA, every type of cellular RNA is transcribed using DNA as a template. And so for each one of these RNA transcripts, each one of these RNA sequences, there is a DNA sequence that allowed it to be created in the first place. Some of these RNAs are transcribed in both prokaryotes and eukaryotes, and these are the major players in gene expression. Some are only produced in eukaryotes, and so these are the smaller regulatory RNAs that I mentioned on the previous slide. And then one exception, this one is only produced in prokaryotes. And we'll come back to the idea of what CRISPR does and what it is in later modules. So all cellular types of RNA are transcribed using DNA as a template. There is, of course, one exception to this rule, which is some viruses can copy RNA directly from an RNA template. But this is very unique and only for RNA viruses, and you only find this kind of phenomenon in a virus. Some other features that are common to all types of organisms, so prokaryotic and eukaryotic, are features of the template, so the template DNA. In the DNA, there is a strand that is transcribed or used as a template to make RNA. This is only one strand The other complementary strand is not used to do this. I'll come back to this towards the end of this video. Also contained within the DNA is a transcription unit consisting of a promoter, which helps to start the process of transcription or creating RNA from a DNA template. There's the RNA coding region of the transcription unit, which is the information in the DNA that encodes for the protein. And then there's a terminator in the DNA that signals to the RNA polymerase when to stop transcribing RNA. A transcriptional unit includes, like I said, a promoter, a coding region, and a terminator. So let's take a closer look at this transcription unit. As I also mentioned, there is a template strand, which is actually used as the template to create the RNA transcript. And there's a non-template strand, which is not used to create the RNA transcript. The template strand is the 3' to 5' directional strand. RNA transcripts are created anti-parallel to their DNA template. And so notice that the RNA transcript that appears here from 5' to 3' would be the complement to the template strand, which runs 3' to 5'. This also means that the RNA transcript running 5' to 3' is sequentially identical to the non-template strand, which also runs 5' to 3', except this one would contain thymines and this one would contain uracils. The promoter region of the transcription unit is where RNA polymerase first binds to start transcription, and the start site is the position, the nucleotide position in the DNA that correlates to the very first starting nucleotide in the RNA transcript. And so if in the DNA it's an A right here in the 3' strand, then it would be a U right here in the 5' strand. Then there's the RNA coding region, which encodes for the actual protein. And so again, you can't read DNA to get a protein, you need to read the RNA to get that. And then we have the terminator sequence, which signals RNA polymerase to stop making RNA, which does get incorporated onto the 3' end of the RNA transcript. When I'm referring to something being upstream, I mean towards the 5' end of the non-template strand. When I'm talking about something being downstream, I mean towards the 3' end of the non-template strand. RNA molecules are synthesized complementary and anti-parallel to the template strand. So let's take a closer look at this now. So here's our DNA double helix, and then right here, there's a transcription bubble that's being opened up by RNA polymerase, and RNA is being created in an anti-parallel direction to its template. So here's the template running 5' to 3'. Here is the RNA running in the opposite anti-parallel direction. It's also synthesized using complementary base pairing. And so if there is a C in the DNA, there will be a G in the RNA. If there is an A in the DNA, there will be a U in the RNA, and so on. New nucleotides are always added to the 3' OH group of a growing RNA chain, just like DNA replication adds DNA nucleotides to the OH group of a growing DNA chain. So therefore, transcription also, just like DNA replication, proceeds in a 5' to 3' direction. As I mentioned before, RNA is transcribed from one DNA strand, not both. And so let's take a look at why this might be. Here's gene A. Gene A is going to have, so we have gene A in the, on the negative strand and the positive strand, right? And so these are complementary to each other. RNA here is being transcribed from the positive strand, from this 3' end. And so we get a 5' to 3' RNA molecule that's anti-parallel to its template. When proteins are made from this RNA molecule, it starts from this end and works its way through the template to create the protein in the right order. If that enzyme were to read this in the opposite direction, it would put in a totally new and different set of proteins, which would make a nonsense protein. It wouldn't be functional. And so RNA can only be transcribed from one DNA strand, ensuring that the protein that's made from this RNA is the correct protein. Genes can be transcribed from either one of them, but they are not transcribed from both simultaneously. So here's gene B. This one is transcribed from the negative strand, but not the positive strand. And again, in transcription, nucleotides are always added to the 3' end of the RNA molecule. One of the things that distinguishes transcription from DNA replication is that transcription does not require the use of a primer. RNA polymerase does not need an open OH group at a 3' end to begin making an RNA strand. Remember that DNA polymerase does require this. DNA polymerase needs an OH group on the 3' end to start adding DNA nucleotides. RNA doesn't need this. So it will bind to its target and then start adding nucleotides that are complementary and anti-parallel to its target. They are added to the 3' end of the RNA molecule, and the DNA will unwind and move downstream and then rewind on itself back up as the transcription bubble progresses.